could answer, it seems like pretty much any question about the brain. So with that, Donna, go ahead.
That's setting me up. Okay, so welcome, and I'm glad you could join us. I'm here to talk about my kind of personal research journey to develop a therapeutic. And I'd like to say that it is a therapeutic for Alzheimer's disease, but it's mostly a therapeutic to prevent neurodegeneration in the brain. That is, to prevent the brain from actually declining or dying because it's been subjected to a disease process or to an injury. So hopefully I'll be able to explain this in terms that are understandable. And if you have questions about the science or the science words that I use, please feel free to ask me at the end of the talk. And hopefully I'll do a good enough job explaining this that it'll be clear. So why is this not actually, hang on. There we go. Okay, so about me, I'm an associate professor at the University of Utah in radiology and imaging sciences. I'm a neuroscientist, and I've been in this field for about, actually more than, I've been in the field for 25 years. And, you know, my goal, I'm originally an engineer. I started out at Michigan as an aerospace engineer, and the Challenger blew up when I was in college. So that career was kind of not really an option anymore. And I, after getting married and having children, I pivoted to neuroscience because I felt like the brain, you know, space, the brain, you know, a lot of things that are a little bit different in those things, but they were problems that are complex, and I felt could be a useful contribution to the world. So I wanted to, at first I just was interested in studying Alzheimer's disease, and that kind of pivoted into diagnostic and therapeutic options. I'm the current president of the Society of Nuclear Medicine and Molecular Imaging Brain Imaging Council. I've been married, as I said, for 34 years, and I'm a mother of 2 grown children. My son Ian is a data scientist and went to the University of Washington, and my daughter went to the University of Utah Medical School and is a resident at Mount Sinai in New York. And as far as my motivation, I also like to garden and bake, and I'm an athlete. But my motivation for getting into Alzheimer's research is, first of all, my grandmother, my grandma Mary, who died from clinically diagnosed Alzheimer's disease 20 years ago. I mean, she may not have had actual Alzheimer's disease, but that's what she was diagnosed with, as well as my father-in-law here, who is currently living at the Ridge on Foothill in their memory care clinic. And he actually does not have Alzheimer's disease. We have determined using brain imaging that he has trauma brain, which is, he has dementia that is derived from a car accident that he had in the 80s. And so I think that pretty significantly early in my career, I was very interested in how something like a brain injury could actually cause a pathological process in the brain that was actually leading to dementia. So that was kind of where I was coming from when I started this 25 years ago. My original research, I was at the University of Michigan trying to get into the PhD program. So I went into the lab of Dr. Satoshi Minashima. He is the first author on this paper that I've now put on the slide here. And he is the current chair of radiology at the University of Utah, which is why I'm here in Utah. He recruited me after we were at I followed him from the University of Michigan to the University of Washington, and now we're here in Utah. And so when I started in his lab in the 90s, he had developed this set of programs for image analysis. So this is a type of imaging. It's called positron emission tomography. You inject a little bit of radioactivity into the patient, and then you get an image of where that radioactivity goes in the body. And in this case, it's a little bit of radioactive sugar called FDG. And that sugar is what the brain uses for activity. So the brain pretty much uses sugar as its food, so to speak. And so we see when we inject this into a subject, we see a map in the brain of where the most active areas of the brain are. And if there's an injury or a disease process, we then can process, this is what his algorithms that he developed did, they process the brain image to extract the information so we can clearly see a pattern, which is this bottom row, of brain activity, or in this case, loss of activity. So these bright colors on this bottom row is actually areas of the brain that aren't working the way they should be anymore. And with this special type of image processing, he discovered that in very early Alzheimer's disease, these are subjects that were imaged with the PET scan, positron emission tomography, we call it PET. When they would later develop dementia, like 5 years later, they'd go back and they'd look at their scans from early on before they had dementia, and they'd say, oh, look at this pattern. So this is the first time anybody recognized that there was actually a pattern of decreased brain activity that was diagnostic for Alzheimer's disease. And it was a very important finding in the history of this research. So this is actually what Alzheimer's disease looks like. This is a group of subjects who have it. So this is a sideways look at the brain. So looking side on, and these are the areas of the brain that are decreased in Alzheimer's disease. But the very important area is this posterior cingulate, that's its name. It's in the middle of your brain. And that's where you solidify your memories. It's your cognitive processing. Every single day you live your life. And this is where your brain actually kind of creates those memories. So that's why it's very important to Alzheimer's disease. And so basically when somebody gets a PET scan, positron emission tomography, this is what the radiologist who reads the scan looks at. And, it's very difficult for somebody who isn't highly trained to actually make a diagnosis from a scan like that. But when you use these programs to actually process these images, the ones that Dr. Minishima wrote in the 90s, you can easily see, here's the posterior cingulate. It's decreased because that's a bright color, it means down. There's a little bit on the outside, just like we saw in that last image. And this is just a single patient that's been processed this way. So it created a way for clinicians to easily diagnose the disease. And in fact, you can diagnose very different types of diseases. So here's a mild Alzheimer's case. Here's a severe Alzheimer's case. Here's somebody who has a disease called diffuse Lewy body. So this is a patient who has a different disease, and it has a different pattern than you see with Alzheimer's disease. It's similar, but it also has some very unique differences, like occipital lobe area. This is front temporal lobe dementia. Frontal lobe is affected. So we can go through and stratify the various diseases with this simple, very reasonably inexpensive scan, which is called PET. You can diagnose a lot of different types of dementia with it. And in fact, we offer these programs for free. People can download them, and they are distributed to 57 countries all over the world. We also offer help for people for underprivileged, underserved countries to develop their brain imaging capabilities. And Dr. Minishima is also on the International Atomic Energy Association, the UN Commission for helping medical imaging using radionuclides in the world. So it's kind of a passion of this lab, but I just wanted to give the context of where we started with all of this research. So basically, going back to our pattern of Alzheimer's disease, we started to think, okay, why? If you have a pathology in the brain, why doesn't it affect the whole entire brain the same? Why do we have this distinctive pattern with Alzheimer's disease? And so our hypothesis back in the late 90s was because we know that from pathology studies that Alzheimer's disease actually starts in this small area of the temporal lobe called the entorhinal cortex. But all these areas that are affected on the FDG PET scan are where this area is connected to. So you can kind of see the way these long connections are to these areas that we can see on a PET scan are decreased. And so it started us thinking, what is about these particular neurons that are vulnerable to the disease process. And we thought, well, it's because they're really long, they have really long projections. Think about train tracks. You have a city train that runs a couple miles, and then you have, you know, an Amtrak train that will run a couple thousand miles. And if you have a little bit of a disruption in the train track of 1,000 miles, it's going to cause a huge backlog, and it's going to cause some issues in the destination. Whereas if you have a city track that's off, people, you know, they take a bus or they walk. You know, there are a lot of different options for short train tracks, but not so many for the long train tracks. So that is a process that's called axonal transport. And so axonal transport is where things that are made in the cell body of a neuron are transported to the synapse, which is the part that connects to the rest of the brain. And the transport is on these train tracks. They're called microtubules. And you package things into these big vesicles, and they have these motors, and the proteins are polarized so that the motors have a direction that they go. And I'm going to play this little video, which I think is kind of cute. I'm going to try to hit it it's not on the right place. It's coming down from YouTube, and I'm going to just click to it, but it just kind of gives you a little graphic of the process. And it also shows these microtubules assembling and also unraveling, which is really important for what we're going to talk about next with this treatment that we've developed. So hang on, we'll put it to, let's try. One second. Okay, here we go. Microtubules are assembling. This is transport. Okay, so hopefully that gave you a little idea with a graphic of what we're talking about here. So here I am, I'm a graduate student. It's 1999. We had this idea that, contrary to common beliefs, that maybe there's something else going on in the brain for Alzheimer's disease that isn't involving amyloid, that maybe it's these axonal transport processes that have been affected by the disease, and it's actually making the neurons sick. So being imagers, we wanted to invent, or at least develop, a new way of imaging this process. And it turns out it's pretty hard to see something that small using, you know, an image. So we switched to a different type of imaging called MRI, magnetic resonance imaging. And we used a metal called manganese, which has the same properties as calcium. And calcium is a very important ion in the body and in the brain. And so the manganese gets taken up into the brain, and it actually is transported down the axons on the microtubules that I just showed you. And so we can trace it throughout the brain, and we can measure the speed at which it's transported. And we did this in rats through the olfactory bulb. We did a manganese injection. You can see this metal makes the image bright on an MRI because it's magnetic. It's a metal ion. And then we could trace its movement through the brain in rats and actually measure the speed. And we showed, this was my entire thesis, dissertation thesis, that with aging, the axonal transport process gets slower. And we also then again showed it in using transgenic mice who have been genetically altered to have Alzheimer's disease. We showed that the process is affected even when they're very young. So we showed it old age rats, but also in very young mice transgenically altered to have Alzheimer's disease. So hang on. Okay. And one last piece of this puzzle is to kind of try to tie these two imaging modalities together. And this paper, we're going back to humans now again. So these are human images. And we did PET scanning in a group of mildly demented individuals, normal individuals, and people with confirmed dementia. And then we also did MRI imaging to look at how intact those long railroad tracks were. We used a different type of imaging called diffusion tensor imaging, but that's not important. But what we found was if the railroad tracks, if the white matter tracks were degrading, then the FDG PET, the metabolism, the activity in the brain was also degrading. So that was a clear connection between these two different things to say, okay, we're onto something here. This FDG PET that we observed was, the metabolism was declining in Alzheimer's disease patients at a very early stage of the disease. It is definitely affected by the integrity of their white matter tracks that connect the different areas of the brain. So that's when I came up with this idea, and it was pretty early on, like 2011, to say, okay, if we were to take these microtubules, those things you saw connecting and unraveling in the video, if we were to actually stabilize them with a drug so that they don't unravel as easily, will that help prevent the outcomes of the negative consequences of the disease? And so that's what we were looking at, is drugs that are called microtubule stabilizers. And that's when we came up with one that's a common cancer drug called paclitaxel. If you have any loved ones who have undergone chemotherapy, they may have gotten this drug. And it prevents cells from dividing in cancer. So that's why they give it. It prevents them from the tumors from growing. But what we decided to do was to give it in very, very low doses. Oh, sorry. But this drug doesn't cross the blood-brain barrier under normal circumstances. So we decided to give it through intranasal administration. People know that you can give drugs intranasally and they get into the brain. And so that's how we decided to deliver the drug. And this was our original research. It was funded by the Alzheimer's Association back in 2011. And it's a paper where we gave intranasal paclitaxel, very low dose, to these mice who were transgenically altered to have Alzheimer's disease. We looked at axonal transport with our imaging method, but we also looked at axonal transport using cell cultures in vitro just to verify our result. And we found that the drug did indeed normalize axonal transport in Alzheimer's models of decreased transport. We also found, to our surprise, that it influenced inflammation in the brain. And I know that a lot of people hear a lot about inflammation, like related to COVID and a lot of other things, but in Alzheimer's disease, we have a lot of inflammation that becomes part of the process of the disease. So we found that the drug actually reduced the inflammation in the brain. And we also found that it reduced the tau, which is the other pathology. So Alzheimer's disease has amyloid pathology and tau pathology. We found that it reduced the tau pathology in these brains that were transgenically altered for having Alzheimer's disease. However, we didn't see any effect on amyloid pathology. And, we weren't surprised by that finding. When we went to do behavioral tests on these mice, we showed that, so this is a graph of a maze. And so the mice are put in the maze and they have to find their way out of the maze. And this top line are the Alzheimer's mice who were old. They were visibly demented when we started giving them the drug. So these were like 11-month-old mice. And we could say, okay, here are mice that have Alzheimer's disease, and here are wild type mice that are the same age that don't have it. And you could look at them. They didn't look the same. And we just decided to treat them. It was almost a whim. I thought there was no way these very demented old mice who are very sick are going to be able to have any difference giving this drug. I thought we'd have to give it to them a lot earlier, but we did it. And it turns out that when we gave them the drug, even though they were old and had a lot of kind of dementia already, they were able to solve this maze a lot better than the subjects that did not get the drug. And in fact, here's the bottom line are the wild type mice that just learned the maze. And this middle line are the ones that got the drug, and the top lines are the ones that didn't. And in fact, the top line ones, those mice actually just started dying. So we had a hard time keeping the mice that didn't get the drug alive to live, to finish the testing. This was also a maze that we did that tests anxiety, and we showed that the drug also treated anxiety in these very old and demented mice. So that was the result of that study. We then also, I told you I was very interested in brain trauma and brain trauma's effect on the dementia process. And so we then decided to give the drug to mice with brain injuries. And our injury model was a repeated concussion model like you would get in, say, football, where we did 5 mild concussive injuries 5 days in a row, and we gave the drug. And interestingly, this is a lot of science stuff on the slide, and I'll just try to condense it for you. This image right here, this is an MRI image. And all it's showing you is that the mice that didn't get the drug all had these microbleeds under the area where they got hit. So this is the area where we actually impacted the brain. And these are the mice that didn't get drug. And the mice that did get drug didn't have a microbleed. I don't know why. I still have to figure out why we prevented a microbleed by giving the drug. That's a subject of further research. But we also showed, going back to, we did, again, FDG PET, that's the metabolic scans on the mice. And we showed that the mice that got hit had a decrease in their metabolism, and then the mice that got the drug were back to normal. And these are the sham mice. These are the mice that didn't get hit. They're basically our control group. And then we also did pathology, where we showed that there was no axonal injury if the mice got the drug. And we also showed that the synapses, the ends of the neurons, were preserved if they got the drug. But most importantly, and again, I go back to this often, the most important thing is that the mice that got the drug were able, these are our TBI mice now with the maze. This is the graph of the maze, and this is just a little different presentation than I showed you before. But essentially, this gray bar are the mice that got concussive injuries and didn't get drug. And then the white and the black is the controls, and the white are the ones that got the drug. So again, we've mitigated brain injury by giving this drug. So what is the summary of the findings from these early studies? We found that we completely restored or rescued memory with our maze. We improved anxiety with the elevated plus maze. And then as far as inside the brain, we showed that we've rescued axonal transport, we've prevented axonal injury, we've prevented the FDG PET, the metabolic brain activity, from being decreased, and we've maintained synapses in these mice. With Alzheimer's pathology, we reduced the tau pathology, but we didn't do anything to the amyloid. So, the drugs now that are targeting amyloid, that are seeing such a very meager effect, maybe it doesn't matter if you reduce amyloid. If you do all these other things to improve the brain, you restore cognition, maybe that's what's important. And we don't care so much about what the amyloid is doing in the brain at a certain point. We also showed, surprisingly, that we've reduced neuroinflammation in these brains, and we think that's pretty important for future research. So what are we doing now, and what did Alzheimer's Association fund? Well, they funded this grant. As I indicated from my early research days, I am not a drug developer. I just came up with this idea and kind of got sucked into this whole career path. So I've been kind of, getting through with what I've been able to learn. But luckily, in my building at the University of Utah are these very super famous drug development delivery people. And Henry Kopachek is world famous for drug development, but his space is cancer. They haven't worked in brain therapeutics before. And so I just kind of sent him an email one day. I didn't realize he was even in my building. And it turns out we're in the same building. And it turns out my building is named after his lab. It's the biomedical polymer research building. And that's his lab. So they are very hardcore, and they know what they're doing to get a drug from an idea, which is where it was at with my previous research, to clinical trials, which is where we're headed with this. And what the Alzheimer's Association funded was something called pharmacokinetics, biodistribution. And we're going to do all of this so that we can apply to a larger grant to really put this into a clinical trial. So what is pharmacokinetics and biodistribution? It's not particularly scientifically fun, but it just means when we inject the drug into a mouse or a human, where does it go? How much gets there? How long does it stay? So but we have to know these things in order to do a clinical trial, and they're super important. So that's what we're working on with them. Basically what they've done, so they've taken paclitaxel, which is the generic drug I worked on intranasally in the previous studies, and they've attached a polymer and a peptide to it that changes the properties in the body. And these are really important changes, because what the polymer does is, if you inject the drug intravenously, the polymer prevents the drug from being cleared into the liver. So it keeps it in circulation. What does that ultimately mean? You can reduce the dose. So we know that when we give this dose at cancer dose, when they give this drug at cancer doses, it is toxic in certain levels, and we don't want to cause any toxicity in the brain. So we want to give a really low dose. And this polymer allows us to reduce the dose even more, because it doesn't get cleared by the liver as fast. It stays in circulation. What does the peptide do? Well, if you remember from what I told you before, the drug doesn't get into the brain. That's why we were administering it with intranasal administration before. But with the peptide, it actually acts as a chaperone and takes it into the brain. And once the drug with the polymer and the peptide are in the brain, those little pieces are cleaved off, they're degraded, and just the drug remains, and it goes and does its job. So we've been able to reduce the dose on this drug to 1/30th of a cancer dose, as well as increasing its efficacy and reducing the side effects. So, I mean, it's actually just completely incredible how revolutionary these ideas are for drug delivery that Henry and Jane have come up with. So this is just some data to show that when we inject the drug into the mouse, it gets into the brain. That's pretty simple, and that's awesome. And we're going to do more of these studies in the upcoming weeks with the Alzheimer's funding, because this was just kind of like a single mouse, and we're going to do it with a group of mice, and we're going to show how much gets into the brain, how long it stays in the brain, those kind of interesting things. But this is the most important part. I showed you this graph before. This was the intranasal administration of the generic paclitaxel. And we showed that the mice didn't learn the maze, mice that got the drug learned the maze, and then these are the wild type. This is the new graph. And this is just, I have to tell you, I actually texted this graph when I did this data analysis to some of my colleagues, and they were floored. Everybody got really excited. So these are the mice that didn't get the drug. And then the dotted line are the wild type, and the green line are the mice that got the drug. So essentially, there is no difference between the mice that get the drug and the normal wild type mice with this new version of the drug, and a very, very low dose. And this is so exciting. And this is the graph that went into the grant. And I think this is why it got funded from Alzheimer's Association, because it is truly exciting. This is actually different data from traumatic brain injury, where we tested this drug out in my mice that we give the concussive injury to. And in this case, we show, this is just a different way of showing it. This is short-term memory. This is long-term memory. It's still the maze. And it was kind of funny because we got this result. These are our different versions of their drug. This is the generic. This is the sham. These are the mice that didn't get the drug. You can see all these little groupings. But when we gave their new drug, the mice that got the drug did better than the shams. So here are the mice that didn't get hit at all. These are concussive injury mice, right? And I was like, well, how can the mice who get the drug do better than the mice who didn't even get a brain injury? Turns out that the anesthesia that we use to put them under to get hit, we also give it to the sham. It causes a little inflammation. This is back to my inflammation idea. And so there's a little bit of residual inflammation when we do the maze testing that makes even the sham mice a little stupid. But when we look at mice that never had anything done, that's over here, this column, naive, we see that basically these are mice that didn't receive any anesthesia, anything at all. They just basically came in and ran the maze. So that's basically what the drug does. The drug makes the mice even better than anything because it's reducing the inflammation. So that is, in a nutshell, what we've done so far. This is a picture that I take. I live in Park City. This is a picture I took on a walk one day. And I like to talk about science philosophically. So sometimes in science, we have this path that we see in ahead of us, and we have a goal, which is those mountains in the background that we want to get to. But in the middle, there's all this like mist and fog. And you might get into the middle of the mist and might lose your way, or you might find a new path, but eventually those are opportunities for us to find something new. And I feel like that's what we've done with this research. We've kind of followed this path from imaging observations to treatment development. And now we're in a really exciting phase, because I feel like we're in the foothills of these mountains, and we're gonna reach the summit in the next, like, probably 3 to 5 years. And that is thanks to the Alzheimer's Association. And I'm really grateful to them, because they really got us, on this path, getting into the foothills of these mountains. And yeah, so that's my talk. This is our lab. Myself, Dr. Minashima, Dr. Ansai, who is actually Dr. Minashima's wife, but she's also a neuroradiologist who's been very instrumental in a lot of this work. My tech, Max, and all my students and my collaborators, both here and back in Seattle. So with that, I will end, and I'd be happy to take any questions.
Thank you so much, Donna. If you have a question, feel free to unmute yourself or put it in the chat and I'll read it.
Hopefully it wasn't too much scary science stuff.
I think you probably made it as simple as you could be. I could have,
I tried, but don't. There are no silly questions. I'm happy to answer any kind of questions you have, even if it's sort of basic. And if I can, I can't say I can answer everything, but I'll do my best.
Raven, go for it.
Hi, Donna, Dr. Cross. My name is Raven Albertson. I'm the program director with our chapter, and I really enjoyed your presentation. I get approached often when we're out in the community, and people just ask me, like, is this, they just come up with different treatments that they've heard of, and like, should I be doing this or that? Based on the research that you've done, it seems like just reducing inflammation would be inferred as something that we could say to
Well,
people.
yeah, it's amazing to, I refer to the study that they did in the UK, where they showed that lifestyle factors are, can reduce your chance of developing dementing disorders by 30%. So, eating healthy, not smoking, not drinking in excess, and, moderate to moderate exercise. And all of those things, well, the healthy eating and the exercise are definitely anti-inflammatory. Particularly certain foods. And I myself am a huge proponent of both. So I think that if there's anything that anybody can do, and 30% reduction, that's a huge amount. 30% reduction in your chance of developing Alzheimer's disease or other dementias in 10 years, just by leading a healthy lifestyle. So, I mean, that's within our power. And I understand that, it would be great to have a treatment, and we're working on that, certainly 100%, because diet and exercise aren't going to fix everything. But I think that for now, until that day comes, that is what we need to encourage. And by healthy, there are so many inflammatory foods. There's so much out there about the type of things you can eat and the type of exercises and lifestyle changes that you can make. I mean, nobody wants to hear that, but that's the reality as far as I'm concerned with this. Yeah.
Thank you. I mean, I know some people don't want to hear it, but others might think, well, that's something I have control over.
Right, exactly.
So there is something I can do. So yeah, thank you so much.
A question that I had after listening to your presentation was, I was wondering if you're aware of any previous studies where drugs have been used that already exist for Alzheimer's or just brain disease in general.
Well, I mean, that's a very fundable area, repurposing drugs. People love to do repurposing drugs. I would have loved to have obtained funding earlier on in this whole entire process for repurposing paclitaxel. The problem with that from my perspective was it didn't get in the brain. So we had to change the administration route. So we were going with the intranasal route. And because of that, with FDA regulations, you have to redo all the toxicology to show that you're not going to like destroy your entire nasal passage by giving this drug this way, right? And the reality of that is, and the reality of therapeutic development is that a toxicology study is about $1 million. And with a generic drug, there's no one's going to make any money. No one's going to recoup that, right? So that's why, particularly, not only is Dr. Kubitschek and Dr. Yang's drug better, but it's also better because it's associated. It's a proprietary drug, so it can get licensed. It can go through all these tox studies, and it will be developed, and it will go to the clinic for that particular practical reason. And I know people don't understand. It's kind of the really depressing part about therapeutic drug development, but that's the reality. It's as much as anybody wants to say, let's repurpose old drugs for a new purpose. If there's any sort of like glitch when it comes to the regulatory approvals, it's not going to happen because that's just not the way the world works, unfortunately. But is there any drugs, particularly for Alzheimer's disease? None that I know of that have actually shown to be effective, unfortunately.
That makes sense. Any other questions?
Is there something in the chat? No. Okay.
No questions.
Okay. Well.
Okay. Well, if that's it, we really appreciate your time. You're welcome to stay for our meeting, but the rest of our meeting, but you definitely don't have to. We really appreciate your time, and it's so fascinating. I know we have a couple of other volunteers who work in brain health and are researchers themselves, so they're excited to hear your presentation.
Yeah, and you have my contact info. I'm always happy to chat with people.